Good day, everyone. We are here in front of you to discuss about photosynthesis. So, what do you mean by photosynthesis? So, photosynthesis is a process that needs light, water, and carbon dioxide in order to create oxygen and glucose or sugar.
So photo, which means light. Synthesis means to build something or create.
So, in photosynthesis, it is divided into two stages. It is the light-dependent reaction which occur in the thylakoids in the Calvin cycle, or also known as Stark reactions, which occur in the stroma, inside the chloroplast.
So there's gonna happen inside the thylakoid. The H2O go to the thylakoid, and and the light absorbed by the thylakoid. There's how oxidation happens. When there's how oxidation happens, the H2O turns into oxygen gas. So when there's how oxidation happens, there's how an electrons and energy are are or release, or there's how remain energy and electrons. So after that, the NADP plus NADP plus gain the electrons inside the thylakoid that that are remain inside the thylakoid. So the NADP plus gain the gain the electrons, and the NADP plus turns into NADPH. So the ADP phosphate, it also gain the energy inside the thylakoid. So the ADP phosphate turns into ATP. So the reactants of the
Light-dependent reaction.
light dependent reaction are H2O, NADP plus, and ADP phosphate. And also the product is oxygen gas, ATP, and NADPH. So my partner, we're gonna tackle about what happens inside the Calvin cycle.
So now I'm gonna tackle about Calvin cycle. So I can say that Calvin cycle, it's a process of reductive. So when we say reduction or reductive, it is to gain the electrons. So CO2 will go to the Calvin cycle and turn into sugar and to create sugar. So carbon dioxide need electrons and energy in order to create sugar or glucose. So NADPH will oxidize and turn into NADP plus. So there's an oxidation happens. So when you say oxidation, the electrons of NADPH will remain in the Calvin cycle. So those electrons will get the carbon dioxide and help to create glucose or sugar. So next is ATP release. ATP will release the energy in the Calvin cycle, and ATP will turn into ADP phosphate. So since ATP to ADP, which means the release of energy, then the energy which release the ATP will use in the Calvin cycle to fire to make the process ongoing. So the ATP, so the energy being released of ATP will help to convert the electrons and carbon dioxide into sugar or glucose. So the product of this cycle, the reactants of this cycle is ATP and NADPH and also the CO2. And in the product are sugar, NADP plus, and ADP. So that's all. Thank you.
Now I'm gonna tackle about light dependent reaction. So I'm gonna dig deeper the inside the thylakoid membrane. What happens in the thylakoid membrane? So as we can see, there's about pigments. There's about two pigments that called photosystem 1 and the photosystem 2. As you can see in the photosystem 2, there's about constant 2 electrons. So the 2 electrons, or the photosystem 2, are absorb absorb light to become the electrons gain energy or get excited. So the electrons, when electrons get excited, the electrons leave chlorophyll and go to the plastoquinone. So so since the chlorophyll are lost lost electrons, there's have happened the water or H2O split split and the electrons of the H2O go to the chlorophyll and the hydrogen ions go to the inner thylakoid. So there's a remaining oxygen atoms. So after that, we can go back to the electrons inside the plastoquinone. So the electrons inside the plastoquinone are continued go to the cytochrome B6F complex. The energy of the electrons are are used to pump the hydrogen ions hydrogen ions across the thylakoid membrane into inner thylakoid. So since the since the hydrogen ions across to the thylakoid membrane into inner thylakoid, there's have high concentrations happen. So the hydrogen ions are spread in the inner thylakoid, and the stroma are low of concentration. The the hydrogen ions in the inner thylakoid are go to the ATP synthesis. So this is a ATP synthesis. Then ATP synthesis are are the or the hydrogen ions when they across the ATP synthesis, the ADP turns into ATP. But since there's a remaining hydrogen ions, hyd, remaining hydrogen ions, the hydrogen ions are go or spread to the stroma. So this then then the ATP is a K product of a light dependent reaction. So this ATP is a K product of a light dependent reaction. So we can go back to the electrons inside the cytochrome B6F complex. So after that, there's a remaining energy of electrons. So the electrons inside the cytochrome B6F complex are continue go to the plastocyanin. Then the electrons are also continued go to the photosystem 1. But since the electrons traveled, they lost energy. So in in their in there is they need energy from the light. So photosystem 1 are absorbs light to become the electrons become get excited. So after that, the electrons get excited. The electrons continued go to the fluorodoxin. Then after that, the fluorodoxin electrons, or the electrons of fluorodoxin are accepted from the NADP plus, and it's called reductase. But when the NADP plus accept the electrons, they also accepted the hydrogen ions. So this is then after that, when they both accepted the hydrogen ions and electrons, the the NADP plus turns into NADPH. So this is the NADPH is also a K product of a light dependent reaction. So as we can see, there's have oxygen atoms in there. So we need to repeat the cycle to become the oxygen atoms become oxygen gas. So there's have the process. So what I can say earlier, we can repeat the process that the the oxygen atoms become oxygen gas. So the process is repeated. Then the oxygen atoms are go are go to the or added to the oxygen atoms, and oxygen atoms equivalent to or become oxygen gas. So the reactants of the photosystem, or the reactants of the light dependent reaction are H2O, H2O, NADP plus, or ADP. So the product is oxygen gas, and NADPH, and ATP. So, and that's all. Thank you.
So now, I'm gonna dig deeper about the processes happen in the Calvin cycle or in the dark reaction. So, in Calvin cycle, it is divided into three stages or three parts. First, it is the fixations of carbon dioxide or CO2. Second is the reductions of the PGA to G3P. And the third is the regenerations of RUBP from G3P. So first thing's gonna happen is carbon dioxides enter into the cycle or into this process. So CO2 reacted with the RUBP or ribulose 1.5 biphosphate and catalyzed with the enzymes called Rubisco and turned into 3-phosphoglycerate. So that's what will gonna happen in the fixations of the carbon dioxide or CO2. So we have three molecules of RUBP, and each molecule it contains of 5 atoms in 2 phosphate. So carbon 1 contains 1 phosphate group, and carbon 5 also contains 1 phosphate group. So since we have three molecules of RUBP, we're gonna use also three molecules of CO2. So in CO2, one CO2 is equivalent also on one carbon atom. So when one molecules of CO2 reacted with one molecules of RUBP, it will initially result into total of 6 carbon atoms and 2 phosphate. So like this. So but it is divided into two. It is divided into two or broken down into two pieces. So PGA will become PGA will become or contains 3 carbons and 1 phosphate because it is divided into two. So it is a result. So the result is it will become two pieces of 3-phosphoglycerate. So it is called 3-phosphoglycerate because 3 stands for 3 carbons and phosphoglycerate stands for phosphate. So we're gonna track how many carbons being used in this particular process. So stated I stated there that we have three molecules of RUBP and contains 5 carbons. So 5 times 3 is equivalent of 15 carbons. Since we use 3 carbon dioxide, it has also 3 carbon atoms. So 15 plus 3 is equivalent into 18 carbon atoms. So 18 carbon atoms, since in 1 PGA, it contains 3 carbon. So we're gonna divide 3 into 18 carbons. So it is results that we can get 6 pieces of PGA or 6 pieces of 3-phosphoglycerate, 6 pieces of 3-phosphoglycerate. So that things will gonna happen in the fixations of carbon dioxide. So next, we're gonna discuss in the second part. It is the reductions of the PGA to G3P. So as you can see, it has a kinase, PGA kinase. So PGA kinase is used to catalyze the phosphate and turn it into molecules. So in this process, it requires ATP. So we're gonna use 6 ATP to phosphorate the 3-phosphoglycerate in order to gain this result, 1,3-biphosphoglycerate. So I say that PGA kinase is an enzyme that catalyze the process of this and turn into biphosphoglycerate. So ATP will release his phosphate and turn into ADP. So the phosphate being released of ATP will be transmitted into this molecule. As you can see this. Then since ATP gives electrons on this part, it will turn and look like this. So it is called the 1,3-biphosphoglycerate. So it is called biphosphoglycerate because it contains of 2 phosphate. So next stage is we're gonna use the NADPH. So in this case, always remember that PGA kinase is catalyze the phosphate to make it into the other molecules. So, next is we're gonna use NADPH used to convert 1,3-biphosphoglycerate and turn into G3P. So, NADPH will be catalyzed by the G3P dehydrogenase. It is an enzyme that catalyzes the NADP that removes the hydrogen. So, since iyang gi-remove ang hydrogen sa NADPH, then it also removes the phosphate. It also involves the removal of the phosphate of the biphosphoglycerate. So what will happen in biphosphoglycerate, it will turn again into one phosphate, but NADPH give his electrons in order to make the biphosphoglycerate turn into G3P or glyceryl dehydrophosphate. And since we use 6 NADPH and we convert 6 pieces of 1,3-biphosphate biphosphoglycerate, it also results 6 pieces of G3P. So 6 pieces of glyceryl triphosphate. So that's what gonna happen in the second stage or second part in the reductions of the PGA to G3P. So now we're gonna tackle about the next, and it is the regenerations of RUBP from G3P. So since we have 6 G3P, the 1 G3P will go down and it is used to make or to create a glucose. So the remaining 5 will go down and it is called regenerations and turn into RUBP. So since, as you can see that 5G3P contains also 15 carbons, a total of 15 carbons, and uh, I stated there first that the constant molecule also contains 15 carbons, but 5G3P is he needs more energy or phosphate in order to reduce it or convert it into 3-RUBP. So we need ATP. So ATP gives the phosphate or energy into 5,3-GP in order to convert 5,3-GP into 3-RUBP. So that's what will happen in the process of Calvin cycle. The rotation is like that. So since stated there that glucose or sugar are created with 2 G3P. So we need another process in order to get one another G3P in order to create glucose. So that's the order that Calvin cycle will create a glucose in that case. So that's all. Thank you.